Hey everyone, my name's Mac. I'm really sorry I can't be there in person, but today I'm going to walk you through some recent work that we've been doing in my lab at the University of Sydney. And in my lab, we're really interested in understanding how the different arms of the ascending arousal system that project out to the rest of the brain can shape and constrain the types of dynamics, the complex adaptive dynamics that arise in the rest of the brain. In particular, we're really interested in a little tiny area in the pons called the locus coeruleus. Now, this is the main noradrenergic projection to the rest of the brain, and it actually has these massive widespread projections that cover really the entire brain in these little axonal projections that release a neurochemical, noradrenaline or norepinephrine if you're in North America. And we're working under the assumption and testing the hypothesis that through these different projections and the release of noradrenaline, the locus coeruleus can affect complex adaptive brain network dynamics. Down at the microcircuit level, down in at the level of the individual synapse, the ascending arousal system and noradrenaline work a little differently than the typical way that you think about neurotransmitters. So in this cartoon here, the little green circles represent something like glutamate, and you can imagine an action potential coming down this presynaptic terminal, causing the little vesicle of glutamate to fuse with the synapse. It's going to release glutamate across the synaptic cleft. It's going to hit different glutamate receptors like AMPA receptors or NMDA receptors that are then going to cause an action potential to propagate in the postsynaptic potential. In contrast to this mechanism, neuromodulators actually work a little bit more like a hormone in that they are released from the site. They contact a different type of receptor we call a G protein coupled receptor. These little curly green and red receptor cartoons. And through conformational change and internal second messenger cascades, they actually release chemicals like calcium from inside stored vesicles in the endoplasmic reticulum, and also change the expression and the activation of different voltage-gated ion channels. The ultimate impact of this conformational change within cells isn't necessarily to pass an individual spike on in the sequence the way we typically think about a message being passed. Instead, it changes what we call the neuronal gain. And so in cartoon form, what we're thinking about here is how much bang you get for your buck. If an individual input comes into a cell, we'd like to know what's the likelihood that that input will actually get translated into an output. So if there's an input current, what's the firing rate Q that's going to come out the other side? Well, the idea is that neuromodulators like noradrenaline and acetylcholine, serotonin, dopamine, etc., can alter the likelihood that that spike gets converted into an output, or the receptivity or the excitability of the cell. And we represent that with these typical gain curves, which essentially tell you, like I said, how much bang you get for your buck. Now, the typical analogy that we would use in this case, and it's been really quite useful over the years, is to think of the ascending arousal system as affecting something like the volume knob on its target regions. So you can imagine that if the volume knob on an old school radio was turned all the way down, you wouldn't really notice any inputs at all. It doesn't matter what is coming into the cell, it's not going to get translated into an output. In contrast, if I turn the volume all the way up, that's going to be a little bit like when someone turns a microphone up too loud on a stage. All of a sudden you start hearing all this feedback and you get this really awful kind of pooling of all the noise getting propagated through the system. Whereas right in the middle of those two is some kind of a sweet spot. And so people would use this to kind of argue for this nice inverted U-shaped curve you would typically see with studies of neuromodulation. But there are some problems with this analogy that kind of limit our ability to really communicate what we think is the sort of real beauty and elegance of the ascending arousal system. And so a couple of years ago, a really brilliant grad student in my lab, Gabriel Weinstein, came to me with a really lovely update to this analogy, which is to think of the locus coeruleus as something like the bow to the brain's violin. So you can imagine that if you've got a particular, so this is a picture of Gabriel here. So you can imagine that now if we think of the cortex as sort of controlling what we would call the sort of neural melody, the particular sequence of activity patterns that we're going to play out to, let's say, identify an object or a face, or to have a conversation, or to drive your car. You can imagine now the locus coeruleus is going to change the way in which that particular grouping or that particular sequence is expressed. The same way that a bow helps to extract a note from a violin. It doesn't actually change which note you'll play, it actually just tells you whether or not that note will be expressed for you to hear. So you can imagine during sleep, when the locus coeruleus, slow wave sleep, when the locus coeruleus is relatively quiet, what you're going to have is essentially no melody being played whatsoever. So you could have up and down states in the cortex, but they're not expressed at the behavioral level. As we transition into quiet wakefulness, now we have an increase in the relative firing rate of the locus coeruleus, which is now going to imbue certain parts of the rest of the brain, let's say certain sub-networks within the cerebral cortex. It's going to imbue them with that increase in volume so that you can hear the melody being played. Now, if you wanted to become more active in waking, you wanted to do something more complex, maybe now you need the locus coeruleus and more of the locus coeruleus to become active, to now bring on these different melodies across the different networks of the brain. And then you could also imagine if something salient happened, then all of a sudden you'd have a big increase across the entire locus coeruleus, which is then going to douse the rest of the brain in noradrenaline and then change the types of firing patterns and the types of communication events that can occur. So I want to bring this idea of the locus coeruleus and its impact on the rest of the brain into sort of sharp contrast with another really, really common, really highly conserved and very influential neuromodulatory system, which is the cholinergic system. And so the main projection of the cholinergic system to the rest of the cerebral cortex comes from the basal nucleus of Maynard. It's a little small smear of cholinergic cells that sits right underneath the nucleus accumbens in the basal ganglia and right above the hypothalamus, so right in that forebrain area. And in contrast to the way that the locus coeruleus projects out to the brain in a really diffuse manner, the basal nucleus of Maynard projects in a much more targeted, or what we would call a segregated fashion, to the rest of the brain. So to kind of to stretch our analogy quite thin, but I think just to give you that intuition, you could think of the locus coeruleus as acting like a bow, which is going to change multiple networks at the same time, whereas the basal nucleus of Maynard will be a little bit more like pizzicato, where you could pluck a particular string and get that note out without affecting any of the other strings. And so in some previous modeling work, really brilliant postdoc in my lab, Eli Muller, created what's called a neural mass model to try to understand the impact of changing these kinds of broad, diffuse projection patterns onto a working model of typical brain dynamics. And to cut a very long story short, what Eli was able to show was that as you increase the diffuse coupling in the system, as you increase the influence of something like the locus coeruleus, what you effectively do is what we'd call flatten out the energy landscape of the potential states that the brain could reach. So you can imagine that if the value is really low, you're sort of stuck doing whatever it is that you're doing. And as you increase the diffuse coupling, now all of a sudden the potential options are available for you to move around that state space into really novel configurations. So now what we can do is we can think, well, if the locus coeruleus is a little bit like that diffuse system, that ought to give us this flattening. And if the basal nucleus of Maynard is more like that targeted system, that also ought to give us a deepening in this attractor landscape framing. We should be able to test these ideas using neuroimaging data. So another really great postdoc in my lab, Brandon Munn, was really attracted to this idea and wanted to test this in empirical data. And so what he did was he took some 7T resting state fMRI data that we'd received from Luke Hearn and Luca Kochi, who are up at the University of Queensland. We essentially took the cortical resting state dynamics from each of these different subjects. We had 59 subjects in total. And what Brandon did was he quantified how likely it was for a particular brain state. What I mean by that is the configuration of different activity or bold patterns across all the different ROIs we used in a study, how common that different state was. And we defined the likelihood of being in that state as 1 over the probability of that state being present across the resting state session. And then what he did was he extracted time series from the locus coeruleus and time series from the basal nucleus of Maynard. Now, we were very careful to correct for physiological noise as well as other parameters such as the nearby fourth ventricle, which sits right behind the locus coeruleus, which we wanted to ensure didn't affect our results. And what Brandon was able to then do was to take this energy landscape framework, and he was able to then perform a cross-correlation essentially with the locus coeruleus time series and the basal nucleus of Maynard time series. And to cut a long story short, effectively what he found was that there was evidence for our hypothesis that after peaks in the locus coeruleus, after the bold went up in that particular subnucleus, the energy landscape became relatively flat. That meant that it was much easier for the brain to shift into really novel states. Whereas if the basal nucleus of Maynard had a peak in its bold response, you actually found exactly the opposite, that you were much less likely to move to a new state. You were much more likely to stay in the same state. So we found that really, really exciting, but we really wanted to try to understand what the functional benefit of such a pattern would be. And so Brandon found a really lovely data set online where individuals pushed a button while they were doing breath awareness meditation. And then what we could do is we could orient, so that what they had to do was attend to their breath. Obviously, over time, you'll become distracted. You then become aware of the fact that you're distracted, and then you push a button. Now, we obviously can't understand or really tap into what's happening while they're distracted. But what we can do is we can orient our data to that button press. And lo and behold, we found that there was a big increase in the locus coeruleus as well as the energy landscape flattening just before that button press, suggesting that your awareness was mediated by this locus coeruleus shift. Gabriel, the grad student I mentioned before, has been really chasing some of these ideas down in different data sets. So in this case, we have a data set from James Dankert at the University of Waterloo, where individuals sitting in a 3T scanner, watching the series of little black and white images shift from one configuration to another. And they have to push a button whenever they notice that the image has changed. And so our prediction was that the locus coeruleus should flatten the landscape and allow you to tell the difference between image 1 and image 2. And so we were able to measure pupil diameter around that change point, and we saw a really big increase in pupil diameter right around that change point, which is consistent with the notion of the locus coeruleus being involved. And he was also, Gabriel was also able to quantify the landscape topography and essentially show that there was landscape flattening right around that point too, which we think is a really, really exciting confirmation and replication of the original result in a really different task context. Another really talented graduate student in my lab, Natasha Taylor, has been chasing down some other really interesting ideas. So here we worked with Fernando Calamante, took diffusion-weighted imaging from the Human Connectome Project, created seeds in the locus coeruleus and the basal nucleus of Maynard, and looked for white matter connections between the two. And what Tash was really interested in asking was, does the strength of connection between those two hubs tell you something about what will happen in their attractor landscapes over time? And essentially what she found was that if you look at the strength of connectivity between those two different hubs in terms of structural connectivity, it has a big impact on just how flat and how deep the landscape can become afterwards, suggesting a real trade-off between these two that is augmented by structural connections between the two regions. And then finally, we're doing some really interesting and exciting causal work with Valerio Zerbi and colleagues in Lausanne in Switzerland, where they actually optogenetically stimulate the locus coeruleus in mice who are lightly anaesthesized, and we can actually recreate these same landscape flattening effects in mice. So we're really excited by this new perspective. We believe that thinking about the locus coeruleus in a much more nuanced way can give rise to a really beautiful set of hypotheses, both about normal function, but also about how different diseases like dementia, ADHD, anxiety, just to name a few, could be related to pathology in particular ways in this subsystem. And with that, I'd like to say thank you very much. Real shame that I couldn't be there in person, but please do reach out. I'm happy to answer any questions and hopefully see you next year.